Welcome to the Plant Reproduction and Defense Lecture. We've got a little roadmap here on what we're going to discuss. Reproduction in flowers is all about coevolution with pollinators. So we'll talk about pollination and animal vectors. We'll talk about self-pollination, when it's good and when you want to promote outcrossing. And then we'll get into some of the other methods for reproduction that are not sexual. Right? So let's just get into it with pollination. So pollination is the process whereby pollen leaves from the anther of one plant. Right, so there's our pollen grains. They go from the anther of one plant to the stigma of another. That's just pollination. Transfer of pollen to the stigma. Alright, there are a variety of methods by which this can be achieved. Wind is one of the classics. So wind is a simple pollination method. Conifers, gymnosperms are all wind pollinated. 65% of angiosperms, however, show coevolution with animal pollinators. So these animal pollinators would be vectors. Living vectors to transfer pollen from the anther to the stigma. Animal pollination is great because it increases dispersal distance. Right? So an animal pollinator can often travel further distances than the wind will carry pollen, thus allowing your plants to pollinate other plants that are even further away. So increases essentially the gene pool available for breeding. Right? So having animal pollinators gives you an increased gene pool for breeding, increased mates. And since flowers co-evolved with pollinators, we have a large diversity dependent on their particular vector or vectors. So there's a significant diversity of pollinators and flowers. So for instance, we have a question here. Why might evolutionary selective pressures work against attracting an insect pollinator? So under what conditions would an insect pollinator not benefit the plant? Give you a second to think. And if we just take a peek right here, we can contrast bird pollinators with insect pollinators. So hummingbirds feed off the nectar of flowers and in the process get pollen on their feathers, so that when they move to another flower, they transfer the pollen to the stigma of that flower. So bird-pollinated flowers have to produce large volumes of nectar. Right? If you look at the amount of nectar needed to satisfy an insect versus a hummingbird, hummingbird comparatively takes a lot more nectar. Right? If you just had an insect, it would get full at a single flower. So it would have no need to move on to another flower and pollinate. So, because we have large volumes of nectar, because those large volumes of nectar would completely fill up an insect pollinator such that it wouldn't need to visit anything else and thus wouldn't transfer pollen, we have adaptations to prevent or at least avoid insect pollinators from finding this plant. So, for instance, red flowers, right? Red is a color spectrum that birds can see. It's visible to avian dinosaurs, but generally not visible to insects. So this is great because your red flower, right, will attract bird pollinators to it, but will look bland to insect pollinators, and it will be less likely to transfer it. Right? So in insect pollinated plants, you often see a lot of yellow flowers. And this is because yellow reflects UV light pretty well, and insects see in UV. So insects can see ultraviolet radiation. So smaller flowers with lower amounts of nectar that reflect ultraviolet radiation to make them more visible to insects are attracted, allow insect pollinators to make use of them. They produce such small amounts of nectar that birds would not tend to pollinate them. Whereas the bird pollinators tend to have colorations that make them unattractive to insect pollinators and attractive to birds, and of course they crank out tons of nectar for their bird pollinators. So, a lot of angiosperms show good old-fashioned wind dispersal of pollen. It was cottonwood season just a little earlier. I don't know if cottonwood seeds are still blowing around, but earlier this past month in June, we saw those cottonwoods going around. The cottonwoods are actually seeds, but interestingly enough, cottonwood pollen is also wind dispersed. So that's pretty cool. So in the spring, the cottonwood pollen gets distributed amongst the wind, and in the summer, the seeds get distributed. So it's pretty cool. Okay, now it's time to talk about self-pollination and outcrossing. Outcrossing is basically avoiding inbreeding. So, trying to mate with genetically less or unrelated plants. That's outcrossing. Self-pollination is just straight full-on inbreeding because you're pollinating yourself. It's actually pretty frequent in a lot of angiosperms, and that's because it's a very stable strategy in predictable, stable environments. So, by promoting self-pollination, you maintain your advantageous traits. It's a great way to maintain adaptive traits, is by self-pollinating. Gets you a little bit of stabilizing selection there. So, it's favored in stable environments. One of the big advantages of self-pollination is that you are independent from pollinators. So you do not have to worry about a pollinator finding you, collecting your pollen, and depositing your pollen on another flower, right? Another thing is that it has a comparatively low energy cost. This is because if you're self-pollinating, you generally don't need a big flashy flower, and you generally don't need a ton of nectar. So, and finally, this is the whole maintains adaptive traits. We have sort of a uniform phenotype where we maintain our adaptive traits. So, great for maintaining your specialized adaptations to the area you live. However, outcrossing is a necessity in a lot of plants. Outcrossing, avoidance of inbreeding, basically promotes significant amounts of genetic diversity. Right? So it promotes genetic diversity. This is good because evolution by a natural selection acts on genetic variations in a population. So, when we talk about trees and sexes and outcrossing, we'll often talk about monoecious and dioecious. Okay, so monoecious. Both the male and the female structures are on the same tree, on one plant. A dioecious plant is where we have separate male and female plants. For instance, this is a male reproductive structure. They call it the catkin for some reason. But at any rate, it's a cluster of pollen-producing structures, like a pollen flower, on a willow. So willow tree is dioecious. Having two separate sexes. So most plants are monoecious. It's only really about 6% of all plants, of all angiosperms, that are monoecious. So, monoecious, on plants you have separate male and female structures in the same plant. Basically, you have flowers with both male and female structures present. So the male and female structures are present on the one plant. Dichogamous, that is a little bit interesting. So in a dichogamous plant, we add some temporal protection against inbreeding. How that works is in a dichogamous plant, the pistil and the stamen mature at different times. This means that when the pollen is ready for dispersal, the stamen is not ready for pollination. So it prevents inbreeding and increases outcrossing rates, which is good. And then we have, so this picture here at the top is kind of cool. It talks about some dichogamous flowers based on position, not so much things like season or sunlight or anything, but just position on the plant. So when bees move around this particular plant, it's fireweed, by the way. When bees move around fireweed, they start at the base of the flower stalk and curl around the flower stalk from the base to the top. Right? So at the base, they encounter female reproductive structures, the pistil structures. So down here at the base, we have the pistil. And then up here at the top, we have our mature anthers. So when a bee first lands, it has nothing to deposit on this pistil, so it just goes up around that plant. Eventually it reaches the mature anthers and collects pollen. And the cool thing is the bee comes back down to that flower that is preventing inbreeding. That's cool. Another mechanism to prevent inbreeding is incompatibility, genetic incompatibility. So what this means is that there is a genetic mechanism in place, and that mechanism detects self-pollination, and the cool thing it does is it inhibits the pollen tube growth. So, when the pollen grain can be recognized by the genetic mechanisms involved, there are plants that can activate a pathway that ends up inhibiting the pollen tube growth on a plant that's too closely related. So that's pretty neat. Genetic incompatibility. Probably evolved from systems that recognized foreign incompatible pollen. So, incompatible pollen from plants that are in different species. So, from systems that recognize foreign pollen. So that's pretty cool. Let's see. Let's get on into the less fun part of plant reproduction, and that's asexual plant reproduction, cloning. So we can divide plant reproduction when it comes to sort of self-reproducing into two bits: true asexual / cloning reproduction, and what we call apomictic parthenogenesis. So, apomixis here refers to apomictic parthenogenesis. Apomixic sounded dumb, I guess. Alright, so when we talk about cloning, we are talking about vegetative reproduction. It's very common in harsh environments. You can see it in two basic ways. One, we could have the cloning of parts from the parent cell. Then we could also see runners. Runners are a variety of different potential sources, but basically what you get is something like perhaps a fern that carries a rhizome, underground horizontal stem. So rhizomes can be easily modified. Right, so a rhizome, a modified rhizome to make a runner, allows you to crank out clones of the plant at different locations. Ugh. The more well adapted you are to your microenvironment and the more stable your environment is, the more likely you are to do something like self-pollination or asexual reproduction. A lot of plants in the Arctic undergo asexual reproduction because, well, the Arctic's pretty much the Arctic. So, pretty cool. Apomictic parthenogenesis is interesting because in this we get meiosis. Woo! So a great example is right there on the lawn. Good old Kentucky bluegrass. So the Kentucky bluegrass plant is right there on the forefront of apomictic parthenogenesis. No, the good news is that due to meiosis, we actually still get a shuffling of genes. So we promote a more genetic diversity than cloning. So more genetic. Oh, hello. Wow, okay, that is a great pin. More genetic diversity than cloning. So, this allows us to swap up our embryos and get new combinations of genes, even though we're still producing our own fertilized seed containing an embryo. So, female plants go through the whole process with the megasporangium, megasporocyte, developing into 4 megaspores, only one of which survives, produces the gametophyte with its 2-polar dinucleate tissue, and the embryo. And yeah. I may have gotten lost there for a second. I'm very tired, and I stayed up all night with my dying frog. So, doing great. I'm going to try this over again. Females undergo the whole megasporangium, produces the megasporocyte, which produces the megaspore. Only one megaspore survives to produce the megagametophyte. The megagametophyte produces the egg and the binucleate tissue. The pollen sperm nuclei fertilize the egg and the binucleate tissue, so you get 3 sets of genetic information in the seed, in that binucleate tissue. Now, trinucleate, and what's that called when you got 3 sets? Triploid! That's right. So we have triploid endosperm and a zygote that will develop into an embryo. So we are still asexual. That's an important thing. This is still very much asexual reproduction. It's just we kick in some meiosis in there. So, genetically, these are going to be close to their parents, and they have the advantage of a seed for dispersal. So they can still disperse via a seed, which is important, right? This is a plant they call the mother of thousands, the maternity plant. And basically, it produces these teeny tiny little plantlets that grow to a certain point and can be dropped into the soil around the plant and grow into full plants. Okay, next up we will talk about the different tissue types that we see in plants. We see vascular tissue. So we'll talk about the vascular tissue. We'll talk about the outer covering of a lot of plant tissue, and some plant tissue carries it inside in the anyway, then we'll have the ground tissue. Then we'll hit roots, stems, and leaves. So, without further ado, let's start with the types of tissues. First off is the epidermal tissue, right? So, epidermal tissue, right? It covers the outside of the plant, right? It makes up the dermis, the skin of the plant. Right? So, epithelial cells cover the outside of the plant. Most of them lack photosynthetic pigments. What this means is that you still have to gather nutrients. So when we talk about ground tissue, we'll talk about leaves themselves, but epidermal layers on the outside of the leaves tend to be clear to allow sunlight to the tissue of the leaf. So, a neat thing that we have here are guard cells. Guard cells will appear on either side of a stomata. They flank the stomata. And these guard cells contain chloroplasts. So they are photosynthetic, unlike just about all the other examples of epidermal tissue that I found. So the guard cells are photosynthetic. They flank the stomata, and they allow for closing and opening of the stomata to manage evapotranspiration, to manage water loss. Okay, so the dermal tissue can have specialized structures called trichomes. These are hairs, sometimes fuzz. These trichomes basically help keep the leaves cool. So this is the trichome here. So you get that fuzzy layer around a leaf, and it actually does not make it warmer. It helps keep those leaves cool. And it regulates the climate on a leaf. Right? We can indeed talk about climate at very small scales. So, we call this small scale a microclimate. So, trichomes keep leaves cool. They can regulate the microclimate to keep things optimal. So they can keep the leaf surfaces cool. They can also secrete glues in some of them. You find it a lot in certain carnivorous plants. So, that's pretty sweet. Then we have the root hairs, which is basically hairs or fuzz on the roots. And the big thing here is we are maximizing surface area. Maximizing surface area. So, we increase the efficiency of absorption of water and various nutrients. Continuing forward. The tomato trichomes actually secrete a substance right here that will glue insects to the cell. So it can glue herbivorous insects to the stem, and basically if they can't move around, they starve and die. So it's a good adaptation against herbivory is to have these root hairs that the root hairs that increase all that surface area. I feel like I just went repetitive again. Apologies. I am dying of sleep deprivation here. So, we increase the efficiency of absorption of water and minerals just by having all sorts of little hairy roots. I bet you didn't know that tomatoes had little innate glue traps on them, which is also very cool stuff. Okay, so now let's talk about a little convergence that's kind of cool. This and this are not cacti, which is pretty amazing, right? The needles here are actually modified trichomes. So, they are small. This is a plant belonging to the Brassica genus. You probably know Brassica from Brassica rapa, which is essentially the wild mustard. A lot of domestic plants were derived from wild mustard. Broccoli is wild mustard. Cauliflower is wild mustard. A whole lot of rice you can cook in it. You can cook olives in it. So, what am I cook? Oh man, I am tired. I started thinking about food. I'm gonna leave that in just so you can see what happens when a brain starts to wander. Anyway, Brassica rapa is the source of a lot of domesticated vegetables that we eat, which is kind of cool. Broccolis are selected from the flowers. Cauliflower, asparagus from the stems. Brussels sprouts from the flower buds. So lots of cool stuff there. Anyway, Brassica rapa has these tiny little needle-like extensions that are derived from trichomes. And because they are derived from trichomes, these are not a cactus, even though they have a similar appearance. So beautiful convergent evolution. All right, continuing on to the ground tissue. So the ground tissue is the primary site of photosynthesis. The photosynthetic tissue, by and large, is all ground tissue. So it can participate in photosynthesis, right? In addition to that, it can store various stuff. So we can store chemicals of various kinds in ground tissue. We have ground tissue that serves functions of protection. And we have ground tissue that's involved with regenerating damage. There are very few cells outside the ground tissue that are photosynthetic. Now, when we talk about ground tissue, we'll generally break it into three separate types. Parenchyma is the most common type of ground tissue. So parenchyma, the most common type of ground tissue. So these are where we find our photosynthetic cells. Primarily. These have thin, flexible walls. They allow the plant to bend, right? So it gives it a little bit of flexibility and a little bit of elasticity, and it lets the plant bend if it's bending in the wind or wilting because you didn't water enough. It's the parenchyma that has those thin, flexible walls that allow that bending. In parenchyma, we can also find large storage vacuoles. So storage vacuoles go over here with storing chemicals. So in the case of a storage vacuole, multiple plants have gotten together and multiple cells within a plant generally fuse together to make a large storage cell with a humongous storage vacuole in them, which is kind of cool. So they can store food. They can store starch. And my other favorite category, other. All right. Next up, we have the collenchyma. Almost forgot. These plants that can store stuff in the vacuole, these cells are often responsible for secretions from broken sections of the plant. So, for instance, latex is secreted from plants. It's white. So that's pretty cool. We get latex rubber from it. In general, sap. Right? So you also get the nectar producing cells as being part of the parenchyma, and you get the protein and metabolic waste storage in there. Protein storage, metabolic waste, processing. Oh my. So, sap, resin, two more examples of substances secreted by parenchyma. Next up is the collenchyma. The collenchyma physically supports plant organs. So it provides physical support. A big source of this physical support comes from the polymer lignin. It's just a type of polymer that we see a lot in plants, lignin. Lignin is a wonderful structural support anatomical feature. You find it a lot in celery, right? When you eat celery, a lot of it gets stuck in your teeth because there's this tough tissue that runs the whole length of it. That's actually lignin-rich collenchyma helping to maintain the structure of the celery. So the stalks of celery are almost made up entirely of collenchyma. So again, collenchyma gives you flexibility and some elasticity. Very important in plants. And then lastly, we have the sclerenchyma. And the sclerenchyma are all about lignin. In fact, if I asked you where the main source or where the big source of lignin is in a plant, you would answer that it's the sclerenchyma. It's that much more important. So, lignin gives strength and rigidity. Right? So it will strengthen fruits and it will strengthen stems. In fact, celery is not our only example. Tree bark is actually derived from sclerenchyma. So, tree bark is a specialized outer covering to protect the tree growing, and it is derived from lignin, which is pretty cool. Lignin also gives you the kind of gritty texture of a pear, and the stony nut or shell in cherry pits. So if you've ever tried to eat a cherry with its seed in, you'll very quickly hit the cherry stone, the wall that surrounds the cherry seed. Okay, we've made it through the basic tissues there. Now let's get on to vascular tissue. And I will pretend my one friend isn't calling me because he's the only one who ever calls me. Stop it! Oh my god. Anyway. Vascular tissue is divided into two types, the xylem and the phloem. The xylem is the site, so the xylem is responsible for transporting, right? It transports water, it transports minerals, it transports some inorganic nutrients, right? Anything from the roots to the rest of the plant. And vascular tissue, like if you trace vascular tissue, you're going to trace it from the roots to the leaves, and in those leaves to the stomata. So the vascular tissue, starting at the roots, connects up to the stomata of the leaves and allows for evapotranspiration. The transpiration is when we're doing our gas exchange, and evaporation increases the rate of water loss during transpiration. So, having your stomata open increases the rate of water loss. Now, that does have a happy accident in that the more water loss you have, the more capillary action pulls more water from the roots into the rest of the plant. So, evapotranspiration can accelerate water transport. can, not will, but can accelerate water transport. So, as I said, it contains dissolved minerals and inorganic nutrients like nitrates and phosphates. These are transported mechanically through the vessels. And that mechanical transport is capillary action. So basically, the walls are, or the diameter of each vascular tissue tube is thin enough that water will literally crawl up it, regardless of gravity. Yay, cohesion! Ugh. Woody tissue is xylem. So xylem is at the center of a tree, and as it grows, as it increases in size, it lays rings down, and those rings coming out from the center point all represent new xylem. So the xylem is always sort of in the outermost ring. And the old xylem, the cells all die, and it becomes, in plants that have it, woody tissue. So the wood from a tree is primarily xylem that has fallen into disuse. Right? If you look at a tree, right, there's your rings, each one telling you a little story, right? And then you have your outer layer, your outer ring. And this outer ring is where we have our active xylem. And so these rings here represent old xylem. That's made up of dead tissue, filled with lignin. Okay, so now we'll move on to phloem. Phloem transports sugars and hormones. And I should really call this carbohydrates to be technically accurate. Carbs and hormones. So, transports carbs and hormones basically from the plant all the way to the roots. So this is the food conductance and hormone conductance mechanism for a plant. And again, it operates on capillary action. So, if you expose phloem to the air, a plant will die. Okay, now let's move on to modifications of the roots. So, the main functions of the roots are twofold. They anchor the plant in the substrate, in or on the substrate, on for something like an epiphyte. And of course, they absorb water and all those nutrients I mentioned. We can actually see modifications to the roots for storage of carbohydrates. So a modification for carb storage is a root modified to become what we refer to as a sweet potato or a yam. Right, so this is a modified stem, not exactly a yam there, but it's a modified stem for water storage And these are our sweet potatoes, which are modified stems for storing carbohydrates and sugars. So that's pretty cool. Sweet potatoes are just roots. They're modified roots. Okay, we talked about the roots and the various tissues. Now let's talk about the actual anatomy of a stem. Right, when pointing out the anatomy of a stem, there's some basic anatomical features to know. One, you are comparing your anatomical features to each other. So you look for places where leaves come out. So each one of these is a node, as is already labeled in the diagram. But why not draw? All right. The distance between nodes, amazingly enough, is the internode. Right. The blade, the modified blade of a stem, is our fancy science term for a leaf. So it's a modified blade, and it makes photosynthesis a lot more efficient to have all that tissue around it. Okay, there are a number of things that we can find here. The petiole is present. The petiole is the stalk off of which the flower grows. The leaf. So this here is the petiole. If a leaf doesn't actually have a petiole, if it attaches directly to the stem, that's called a sessile leaf. And lastly, we want to mention our axillary buds, which are important. Axillary buds are located in the crook of a plant. Basically, anywhere you have a lead growing out, we have an axillary bud. So that's pretty sweet. Axillary buds allow for horizontal growth, kind of like an apical meristem, except an apical meristem includes both the shoots and the roots, whereas an axillary bud is a meristem that only grows outward from the shoot. So, both the terminal buds that look kind of like they may be producing a flower, and the axillary buds are meristem tissue. Okay, more modifications of stems, and we will go through these one by one. This is our little roadmap. We'll be talking about bulbs, rhizomes, runners and stolons, tubers, tendrils, and cladophylls. But first, coffee! So let's hit our little road trip here. Boop. Alright. First type of stem modifications are the bulbs. Come back here, you sneaky little. Trying to not be on writing anymore. So these are the bulbs. Bulbs are basically a large bud with leaves. So if you separate out an onion like this, all the layers of an onion are leaves. So these are all modified leaves. And the whole point of a bulb is to allow a little bit of survivability. So oftentimes bulbs are underground to protect them from the winter. So bulbs have a disc for essentially a stem. And because of that, they do not have like a single root that comes off and then branches into other roots, right? So having a primary root that branches off is sort of the standard for most plants. When you have a bunch of roots branching off just sort of individually from a stem, we call these adventitious roots. So, bulbs are generally subsurface, and they protect the plant during winter and basically allow the plant to germinate back to life. So it allows it to live through the winter. It does not get destroyed by freezes because generally bulbs, as long as they're in an environment that they, are well adapted to, they don't get frozen in the ground. All right, on to a rhizome. So the rhizome is an essentially underground horizontal stem. The rhizome also has adventitious roots. And at the nodes, right, we have little scale-like leaves. So here's a scale-like leaf at the nodes. Generally, they're evidence of the fact that these would have been leaves growing out of the stem. So at each node, we have a little scale-like leaf. And then we have photosynthetic leaves growing out of this sucker. So, photosynthetic leaves. And what you see above ground is a whole leaf. So, important to note. Okay, stolons and tendrils. So, runners are essentially, the scientific term for a runner is a stolon. So think strawberry plant. As a strawberry grows, it sends out these little runners. There's like some leaves here. And then along the runners, it starts to grow new plants. Sure, why not? Complete with the leaves and the roots. And it can allow it to very quickly colonize new areas. Right? So that's the runner. Strawberries, and then spider plants, if you've ever had a spider plant. The little cloned plants hanging off the little, those are runners. Those are stolons. So a stolon is a runner, and it's a modified stem for dispersal. It's really effective because it's dispersal without the need for reproduction and seed production. So that's pretty cool. Then we have tendrils, right? So you can see tendrils in things like ivy plants and grapes. These are examples. So basically a tendril is just a little extension of the stem that wraps around whatever it comes into contact with. So the way it grows, when it comes into contact with something, it will wrap around that something, and thus hold the plant in place. A lot of plants that have thin stems that vine around will have tendrils to allow the plant to climb. So these are big in climbing plants. So in peas and pumpkins, the tendrils are actually directly modified from the leaves, but I keep them in the stem modifications because, you know what? Leaves are direct modifications of the stem. So tendrils can be derived from the stem, and they can be derived from the leaves. Last but not least comes the cladophylls. So cladophylls are seen in the cactus. And a cladophyll is a modified, flattened stem, and it is photosynthetic. So a modified flattened stem that is photosynthetic. That means that the needles on a cactus are actually derived from the leaves, right? The big, thick photosynthetic parts of the cactus are modified stems, and the needles are modified leaves. So that's kind of cool. All right, so focusing on the stem modification that is the leaf. The leaf is the primary site of photosynthesis, and it is, because it has to do a bunch of gas exchange, it's prone to evapotranspiration, so we have some protection for it in the form of a waxy, what was that? Cuticle, a waxy cuticle. So the waxy cuticle, which is a watertight and airtight covering, and then to allow that gas exchange for photosynthesis, we have the stomata. So the stomata can open and close to allow leaves to transpire. So let's hit some modifications of leaves. Our roadmap is the bracts, spines, needles, and insectivory. So we're going to be making a fine differentiation here, and I have already screwed it up in my talk about the cladophylls, because needles are for conifers, right? So these are your Christmas tree leaves, whereas spines are for cactus. If you put your hand on a cactus, you feel spine tingling pain. So, yeah. Whereas in March, you still find Christmas tree needles. Oh, And there's some really cool leaf modifications for being a carnivorous plant. But first off, bracts. Right, so here is your pretty azalea, or no, poinsettia. This is your pretty poinsettia. And a bract is a leaf that is essentially colored or floral. So a bract is not petals. So this poinsettia here, these are full-on modified leaves that are just colored. They are not, however, flower petals. It's an important distinction. However, they sort of function similarly. The big giveaway that these are bracts is right here. You can see the veins in the leaves. So that their vascular tissue is set up pretty much the same as their photosynthetic counterparts. So that vascular tissue in there can tell you if you're looking at a bract. Okay, next up is the spines. So this is a bract. Now we have the spine. The spine is a modified leaf, right? It does a number of important things. What kind of environment do you tend to find cactuses in? Cacti? What's that? Low water? Is that what you said? Someone? Oh, yeah, you over there. You said deserts, right? So, what are some big problems in the desert? Yeah. Over there. You said heat, and I believe you said water availability. So, desiccation is a big problem in the desert. Leaves, with all their stomata, are a great source of water loss, evapotranspiration. So, by having a leaf modified into a spine, we have drastically reduced surface area, which reduces evapotranspiration rates. So, great. In addition to that, I don't think you want to eat a cladophil and just pop it into your mouth and chew. These deter herbivory. They are defensive, if you couldn't have guessed that already. Now, a spine is not a needle, and also a spine is not a rose thorn. Thorns, whether it's a rose or something else with thorns, are actually stems, modified stems. So spines are modified, pointy, scary leaves that reduce surface area for evapotranspiration and deter herbivory. Then we have our needles. So, needles are present in evergreens. Now, they are modified leaves, and they are extremely small, so we reduce surface area, and that reduces evapotranspiration, right? So we find these a lot when we have low water availability. So they are all over the mountains because the mountains pretty much require on rainfall to get most of their water availability. So higher elevations also have higher rates of evaporation in the air. So little rainfall, greater rates of evaporation, you want reduced surface area. Another advantage of these is that if you have reduced surface area, you have reduced snow accumulation on the branches. In other words, it's a little easier to resist branches breaking from a late spring snow. When it snows in the late spring or early summer, those wet snows can weigh the trees down, and if it's got a bunch of leaves on it, it can actually snap the branches off. These needle leaves are able to reduce surface area for snow accumulation and take some of that load off. Key thing to note, needles are most certainly photosynthetic, whereas spines are not. It's the chlorophyll on the cactus that's photosynthetic. So, and then we have our modifications for wonderful, wonderful carnivory, which is pretty sweet. There's my ink. Bastard. And then we have our modifications for carnivory. Right? So you find carnivorous plants in basically nutrient poor soils. So you find them an awful lot in nitrate poor or in general nutrient poor soils. Wow, this is working super good, you guys. Nitrate poor or nutrient poor soils. So, because the soils are extremely poor in nutrients, because the soils are not all that great at supporting plant life, we have a variety of plants that have developed modified leaves for getting the nutrients they need in other ways. So, the Venus flytrap, right? Its leaves are modified to close around something like a small insect. So if you were to look, there's actually modified hairs on the inside of these leaves, and if they are disturbed, it causes a closing reaction where the leaves close shut. These little extensions will actually interlace and completely trap the little bug in there, and then it will digest it there. So you find Venus flytraps in acidic swamps because a lot of acidic swamps are nitrate deficient, nitrogen deficient. Whatever, Venus flytrap. So these are all some very fascinating leaf modifications. And of course, kind of fun. When plants attack. Here's my rabbit-resistant lettuce with all my fly trap bits to kill the rabbit. I don't know, man. It was funny at the time. All right. I think I'm going to cut it here, and I will make a separate video specifically for plant defenses. So this is broken up and not two gigantic lecture, or into one, and is not one gigantic lecture.